Welcome to the deep dive. You sent over some really fascinating research today. It gets right into the nitty-gritty of how our cells age.
It does. Yeah, we're looking at this specific thing, this really odd behavior you flagged in the sources.
Which is why older cells seem to be, well, kicking out their own
No,
mitochondria.
that's the core of it. Yeah. Looking at the data you shared, it points towards this expulsion almost of their power generator.
Mitochondria. The cell's powerhouses. I mean, it sounds counterintuitive, doesn't it? Like an aging factory just ditching its generator.
Exactly. So the big question we're tackling based on your materials is why? Why ditch your energy source? And, what does that really mean for us, for aging?
Right, because figuring this out could be huge for understanding age-related problems. And you mentioned these extracellular vesicles, EVs.
EVs. Think of them as like the cell's little cargo pods. They package stuff up and send it out. Cellular mail almost.
Okay, got it. And the twist, from what I read in your sources, is that these older cells are loading these EVs up with mitochondria, like whole mitochondria, and even mitochondrial DNA.
That's the surprising discovery, yeah. They're packaging up mitochondria, including the mtDNA,
Yeah.
and shipping them out in these vesicles.
So the mystery deepens. Is it just cellular trash removal? Or maybe some kind of distress signal? What does the research you looked at suggest? Is it something else entirely?
That's exactly the puzzle. And to really unpack it, maybe you should quickly remind ourselves why mitochondria are so vital to begin with.
Good idea. So fundamentally, they're the energy factories, right? They take food, oxygen, turn it into ATP, the energy currency our cells absolutely need for, well, everything.
Absolutely everything. Muscle movement, thinking, cell repair, it all relies on that energy. They're absolutely essential.
But there's a catch, isn't there? They're not perfect.
No, they're not. As they produce energy, they also create these byproducts, reactive oxygen species, or ROS, free radicals. And these can cause damage over time, oxidative stress.
So the power plant also produces some pollution, essentially. And this gets worse as the mitochondria themselves age.
It does, yeah. Their efficiency can decline, and they can become a bigger source of that cellular stress.
Okay, so crucial, but potentially problematic as they get older. Now, cells normally have a way to deal with damaged ones, don't they? Mitophagy. I think that came up in one of the articles.
I got it. Mitophagy. It's the cell's built-in recycling program specifically for mitochondria. It identifies the damaged ones, breaks them down, reuses the parts. It's quality control.
A vital process. But what happens if that system starts slowing down with age? Because that seems to be the link here, right? To them ending up in EVs.
Precisely. The research you sent over strongly suggests that mitophagy efficiency tends to drop off as we age. So damaged mitochondria start to accumulate.
Leading to more stress, more dysfunction.
Exactly. And this dip in recycling efficiency seems to be why the cell might resort to packaging them into EVs. It's like the cell saying, okay, recycling isn't keeping up. Plan B, eject.
Which, okay, sounds logical on one level, get rid of the broken machinery. But you flagged a potential problem with just tossing them out in these vesicles. What's the issue there?
Yeah, this is where it gets really interesting, and several sources highlighted this. It's the potential blowback. While getting rid of damaged mitochondria sounds good, the mitochondria themselves, and crucially, their DNA, that mtDNA, when released outside the cell in these EVs, can actually trigger an immune response.
Wait, our own mitochondrial DNA, why would our immune system react to that, isn't it?
Ours. That's the key question,
Yeah.
and the answer goes way back, evolutionarily speaking. Mitochondria likely started as separate bacteria engulfed by our ancestor cells billions of years ago.
The endosymbiosis theory, right?
Exactly. So their DNA, the mtDNA, still retains some features that look quite similar to bacterial DNA. And our immune system has receptors specifically designed to spot bacterial DNA as a sign of infection or damage.
Ah, okay. So the cell is just trying to clean house, maybe send out a signal that things aren't right, but the immune system sees this mtDNA package and basically mistakes it for an invader, or at least a major damage signal.
Pretty much, it sets off alarm bells. And this can contribute to that chronic low-grade inflammation we see increasing with age, something the literature often calls inflammaging.
Inflammaging, right. And that's linked to all sorts of age-related diseases, isn't it? Heart disease, neurodegeneration.
A whole host of them. So this process, highlighted in your materials releasing mtDNA and EVs, could actually be a significant driver of that systemic age-related inflammation. The cell's attempt to help might be inadvertently fanning the flames of chronic disease.
Wow. Okay, that's a real double-edged sword then. Trying to fix a local problem, but potentially causing a systemic one. So given this, what are scientists actually doing about it? The sources mention some potential strategies.
Oh, absolutely. This is a really hot area of research. Your sources pointed to several approaches. One big one is senolytics.
Senolytics, like fisetin, dasatinib, those compounds that help clear out senescent cells.
Exactly. Senescent cells, those are the ones that stop dividing but hang around spewing out inflammatory stuff. And they're thought to be major producers of these problematic EVs. So the idea is get rid of the senescent cells, maybe you reduce the inflammatory EV signals.
Makes sense. Target the source. What about working on the mitochondria directly? Some of the research you shared seemed focused there.
Definitely. Mitochondrial therapies are a huge focus. For instance, compounds like urolithin A seem to boost mitophagy, help the cell recycle damaged mitochondria better, so maybe they don't need to eject them.
Ah, so improving the internal recycling system.
Right. And then you have things like NAD plus boosters NMN and R,
Right.
which aim to support overall mitochondrial health and energy production, keep them working better for longer.
Okay. And antioxidants, too. I saw MitoQ mentioned.
Yeah, mitochondria-targeted antioxidants like MitoQ. They're designed to go right into the mitochondria and neutralize that damaging oxidative stress at the source.
So multiple ways to try and keep the mitochondria healthier, reducing the need to throw them out.
That's a big part of the strategy. But there's also interest, as your materials noted, in targeting the EVs themselves, EV modulators.
Modulating the EVs,
Right.
like
Yeah.
controlling their release or what's inside them.
Both, potentially. Researchers are looking at ways to maybe reduce the release of EVs carrying the harmful mtDNA cargo, or even, looking further ahead, engineering EVs to carry therapeutic molecules instead, sort of hijacking the delivery system for good.
Fascinating. Reprogramming the cellular postal service. And what about tackling the inflammation directly, since that seems to be a major consequence?
Absolutely crucial. Your sources mentioned targeting specific immune pathways that get triggered by the mtDNA, like TLR9 or the NLRP3 inflammasome. If you can block those specific alarm systems, you might lessen the damaging inflammatory fallout, even if the mtDNA is still released.
Right, dampen the overreaction. And you also included some more futuristic stuff, gene therapy, CRISPR, how do they fit in?
Well, those are longer-term prospects, obviously. But the potential is there to maybe directly correct underlying genetic defects that cause mitochondria to fail early, or perhaps use CRISPR to actually enhance the cell's own repair or disposal mechanisms, maybe even edit the mtDNA itself, though that's very complex.
It's really quite something thinking about how these tiny microscopic dramas inside our cells connect to the big picture of aging. So for people listening, why is this deep dive into cellular garbage disposal so important?
Because really understanding these shifts gives us a whole new lens on aging itself. It's not just about wrinkles or gray hair. It's about these fundamental processes breaking down or changing inside our trillions of cells. It impacts everything from energy levels to disease risk.
And the research you've brought together today, it's really showing a path towards, well, maybe restoring some balance, helping cells manage things better as they age.
Exactly. It's a quest to, you could say, restore cellular harmony, to understand why cells start ejecting mitochondria, and then find ways to intervene, potentially slowing aging at a really basic fundamental level. This knowledge is paving the way for, hopefully, truly innovative therapies down the line.
So just to recap then, as cells get older, they start shedding mitochondria via these little packages, these EVs. It seems like an attempt to clear out damage, maybe.
It seems plausible, yes, a way to deal with accumulating dysfunction when mitophagy slows down.
But the downside is significant. That released mtDNA can trigger the immune system, leading to chronic inflammation, this inflammaging.
Which is linked to so many age-related conditions, yeah.
And the exciting part is that scientists are actively exploring ways to tackle this from multiple angles, getting rid of old senescent cells, boosting mitochondrial health directly, maybe controlling the EVs or dampening that inflammatory response. Lots of avenues being explored based on the research you shared. It's a very dynamic field. It really underscores how complex aging is, this constant balance between damage, repair, and sometimes unintended consequences, even at the cellular level.
Definitely. So here's something to chew on as we wrap up. Given everything we've discussed about these tiny vesicles carrying mitochondrial parts, how might really understanding and maybe one day manipulating this process change how we approach healthy aging? Could we move beyond just managing symptoms and start tweaking the fundamental cellular mechanisms of aging itself? It feels like we're just scratching the surface. Thanks for exploring this with us.